Hello everyone. It's my great honor to share with you a research work published in the journal iMeta here. The title of the work is Phenotyping, Genome-wide Dissection, and Prediction of Maize Root Architecture for Temperate Adaptability. The corresponding authors of this study are Professor Li Pu and Associate Professor Liang Lu from the Institute of Biotechnology, Chinese Academy of Agricultural Sciences. Maize root system research, hindered by various challenges despite some known genes, can benefit from machine learning integrating high-dimensional data, as shown by this group's past model using high-throughput data to predict early-stage maize height, suggesting its potential for predicting complex root and other maize traits. By analyzing 316 maize lines, we found root trait differences between tropical/subtropical and temperate maize, used root anatomy data with GWAS to identify genes, including FUT5-related aspects, and constructed a high-accuracy machine learning model from root slice traits, providing tools for maize genetic research and breeding. To investigate how maize root system architecture aids in dealing with drought and nutrient uptake, we cultivated 316 maize inbred lines to the 3-leaf stage, analyzed 16 root morphological, 7 weight-related, and 108 microscopic traits via WinRhizo and cross-sectional analysis, and performed correlation and GWAS to find connections for genetic improvement. A study of 316 maize inbred lines reveals significant natural variation in RSA phenotypes, distinct root length and diameter traits between tropical/subtropical and temperate maize, and unique microscopic root region characteristics in temperate maize, suggesting a slender root system in temperate, adapted maize. Analysis of 316 maize inbred lines from the MaizeGeo database shows a weak positive link between seedlings and adult plants' root traits. There are complex correlations among root traits. Total root length relates to some, most root architecture traits have low correlation with plant structure, and weight, root morphology, and root slice microscopic traits are interconnected and can predict maize root architecture. To understand the genetic basis of weight, root morphology, and root slice microscopic traits, we conducted GWAS on maize, identifying many SNPs and candidate genes. In-depth study of trait-related genes, such as NEF101 and DOF24 involved in root nutrient absorption and others affecting root length and structure, shows these GWAS-identified genes are crucial for maize root system development and function. To evaluate gene mutations from GWAS, we centered on the maize FUT5 mutant from the EMS library. Phylogenetic analysis revealed FUT5's high homology across plants and peak root expression, while qRT-PCR confirmed reduced FUT5 expression caused by a base mutation leading to premature amino acid termination. The mutant's different root system structure from the wild type demonstrated FUT5's key role in root development. By analyzing the transcriptomes of the FUT5 mutant and B73 plants, we identified differentially expressed genes, with GO analysis revealing overlaps with GWAS and prior work, and found that heat shock gene changes in the FUT5 mutant affect heat tolerance and hemicellulose biosynthesis, validating FUT5's importance in maize root development, stress response, architecture regulation, and breeding. To explore the evolutionary history of FUT5 natural variation, we studied its genetic variations in tropical/subtropical and temperate maize inbred lines, identifying 8 haplotypes with different distributions. Analyses revealed links between FUT5 haplotypes and root traits, and positive selection on FUT5, highlighting its importance in maize environmental adaptation. Facing the challenge of obtaining root phenotypes in plant big data, we employed a bagging tree model to predict root morphology from root slice microscopic traits, with good results for projected area, and a comprehensive model to find key microscopic traits. The bagging tree model, capable of partially predicting root system architecture using few traits, holds promise for speeding up maize breeding. This study innovatively integrated data and AI, studied 316 maize inbred lines, identified SNP loci and genes, built a machine learning phenotype prediction model, revealed root environment links, overcame the phenotypic identification bottleneck, conducted comprehensive data collection and analysis, and provided support and tools for intelligent crop breeding.